Sure, yeah, we should be able to. I know a few people are still trickling in, but I think we'll go ahead and get started and have some of the speakers start off. And I guess I'll start by introducing myself. My name is Pedro Sampaio. I am a cancer researcher, and I'm also a member of the Taste of Science Houston team. And it really is a pleasure to be moderating this roundtable today. I know we're coming to the end of a difficult, challenging year for everyone. The COVID numbers are still quite scary. We're at almost 70 million cases around the world, more than 15 million in the US alone. But thankfully, we're starting to see a bit of a light at the end of the tunnel. And really, that's only been possible due to the achievements of scientists and clinicians working at the front line throughout this year, who have managed to really do incredible work in record time, often at the expense of their family lives. And that includes really our participants in our roundtable today. And that has led to incredible advances, both in therapies and in vaccines against COVID-19. On the therapy side, we've been able to bring new treatments to the clinic at a record speed, and there's still more than 100 that are being trialed out at the moment. And these include things as different as new antivirals, new immune modulators like steroids that have been a lot of news stories about, and obviously the use of antibodies from the serum of convalescent patients to COVID-19 patients. And I know we'll hear a little bit more about that during our events today. And then on the other hand, obviously everyone's been paying a lot of attention to the recent news on the new vaccines being developed for COVID-19. There's at least 4 or 5 different ones that have shown really promising results, and these span different types of vaccines, from the typical viral particle-based vaccines that we've known about before to these new mRNA-based vaccines. And obviously, everyone is very excited about the recent approval of the Pfizer and BioNTech mRNA vaccine that's just been approved in the UK and Canada, and just now in the US. That sort of gives us a lot of hope for the months ahead. And I know that we'll be hearing a lot about the differences between these types of vaccines and possibly other ones that are currently in development. Obviously, all of these recent investments in the science of COVID bring a lot of questions. We all have a lot of questions, and we know that all of you have a lot of questions as well. And we've received a huge amount of questions over the past few days, and really, really good questions that we can't wait to get into. And we have an incredible panel today of experts that are working on the field, on the different aspects of both the development of new therapies and vaccine development and deployment. So what we're going to do is I'm going to individually introduce each of the members of our round table, and they'll speak to us for a few minutes about some of the work that they're doing in different aspects of both COVID-19 therapy and vaccine development and rollouts. And then we will move on towards the Q&A part of our event based on all of the questions that all of you have sent us throughout the last few days. And we've gotten a lot of them, and we really hope that we can get to all of them throughout the event today. And we really want to encourage, despite that if you have more questions come up throughout the event today, please post them to our Q&A tab here on Zoom or to the chat on Facebook, and we'll make sure we have some time towards the end to ask our speakers the questions that you come up with during the event. But without further ado, I think we'll go ahead and get started with the science. And it's my pleasure to introduce our first speaker, who is Dr. Eric Salazar, who is an assistant professor of pathology and genomic medicine at the Academic Institute in the Houston Methodist Hospital in Houston. And he's working on new types of therapies for COVID-19, and particularly the use of antibodies from convalescent plasma to treat COVID-19 patients. And I'll let you go ahead and tell us a little bit more about that.
Thank you, Pedro, and thank you, Delphine, for inviting me to speak here with the Taste of Science group. It's really exciting to be with you here today, and I'll make it a brief presentation so that we can get on to the many important questions that that have been submitted. So I just wanted to show a few slides to explain some of the work that we've done. And actually, before I go ahead, can everybody hear me okay? Pedro, can you hear me okay?
Yes, yes.
Okay, great, yeah. So yeah, what I have here depicted on this slide here is the on the left is the SARS-CoV-2 virus, which is obviously the cause of of COVID-19, and it's a member of multiple coronaviruses, some of which have a highly pathogenic potential, and certainly SARS-CoV-2 is one of those. And importantly, when you look at the schematic of the virus, what you see is that the surface is studded with these little sort of like blue spikes, and that's the spike protein. So the SARS-CoV-2 spike protein is the focus of tremendous interest worldwide, and it's the focus of vaccines and therapeutics. And why is that? Well, the virus enters the human cell through this spike protein, and the interaction between that spike protein and the host human cell receptor, in this case, the ACE2 receptor, ACE2 receptor. And there are multiple vaccine candidates, including the recently approved Pfizer vaccine, multiple other vaccine candidates that target this interaction between the spike protein and the ACE2 receptor. And essentially, lots and lots of focus on trying to disrupt that interaction, because if you can do that, then you can neutralize the ability of the virus to enter the cell. And it's also the focus of therapies called passive immune therapies. And passive immune therapy has a strong rationale. What it is administration of antibodies against an infectious agent that would give a person immediate immunity, essentially trying to bolster the immune system such that you already have antibodies against an infectious agent. And there's lots of sources. Convalescent plasma is one of these. And what is convalescent plasma? Well, it's plasma from donors that have recovered from the disease, presumably having antibodies and other parts of the immune system that has helped that donor recover from the disease, and you're trying to transfer that to somebody that's fighting the disease. And there are other sources, something called hyperimmune globulins, which is concentrated forms of plasma, monoclonal antibodies that are sort of synthesized in a lab, and you might have heard of these in the news. Regeneron is one of the companies making the monoclonals. Eli Lilly is another, and both of those have gotten FDA emergency use authorization for outpatients. And then there are preparations generated in animal hosts. For example, there are cows, transchromosomic cows, meaning that they've got they're a cow, but they've got a human immune system. And you can essentially harvest convalescent plasma from these cows that essentially has antibodies that look like human antibodies that you can also transmit to patients. And then convalescent plasma has a long history. In fact, the first Nobel Prize in physiology or medicine was given for the use of convalescent serum from horses to cure and prevent diphtheria. So it's got a long history. And the important thing is that it can be deployed very rapidly and is low tech. And what it's involved is you take recovered donors, you need a donor center to collect the plasma, you need to do antibody testing, a blood bank to store the plasma and issue it, and then you need recipients to receive it. And then those recipients can then go on to become donors also. And early on, we put in place two studies, one regarding the donors and one focusing on the recipients of convalescent plasma. And just focusing on the donors, what we learned overall is that only some donors have high levels of antibodies that would make plasma have efficacy against the disease. Those donors with more severe disease have higher levels of antibodies, and the very high levels of antibodies last for about 60 days, meaning we can only collect convalescent plasma within about 60 days after symptom onset. And then moving on to the recipients, just a few points that I want to make so that we can move on to the next speakers and then the questions. When we put in place our recipient study, we put in place a system where we would select the highest antibody level units available for transfusion. And that's depicted in this graph here. Essentially on the x-axis, I've got an antibody level test 1 plotted against an antibody level test 2, and the dots and squares are convalescent plasma units that we collected. And you can see when I plot those against each other, number one, they correlate very well. You can see roughly a line going to the middle of that graph. And number two, in the red boxes, those are the units that we actually transfused in our studies early on. And what we were doing is sorting. And when we use these plasma units with high level of antibody, when you follow patients over time and you match those patients that were not transfused to those that were transfused, based on certain characteristics like the presence of diabetes, heart disease, age, etc., we saw a statistically significant reduction in mortality, the probability of mortality. So the blue line here is those patients that were transfused. The red is those that were not transfused with the high level of plasma with a high level of antibody. So overall, and when we look at all the studies that we've done so far, we found that some donors, when you do efficient antibody testing, you can find units with a high level of antibody. And especially when you give that to patients that are in early disease, and in our data, within 44 hours of hospitalization, the outcome was significantly decreased mortality. And it's important to go back to that slide originally. It's important to point out that other lines of evidence are in agreement with this. Number one, it just makes sense that if you can disrupt that spike protein ACE2 receptor interaction, that you can neutralize the virus. The vaccine focuses on this interaction, and the monoclonal antibodies also focus on this interaction. So what we're doing now is actually giving out plasma as part of an emergency use authorization by the FDA, but also studying these monoclonals in patients in the hospital. And for example, I'm the principal investigator on the Regeneron study for the hospitalized patients, and trying to determine do those monoclonal antibodies also help people in the hospital. So that's the state of our current studies, and I'll leave it there so we can move on to the next speaker and take any questions when it's time for the question and answer portion. Thank you.
Thank you very much, Dr. Salazar. That was really very, very interesting. And I know I have a lot of questions already, but I am going to save them for the end, as you said, so we can go through all of our speakers first before we get into the discussion. And on that note, I'll go ahead and move on to our second speaker of the night, who is Dr. Navin Varadarajan. And I'm really sorry. I hope I said your name properly.
No, you got it.
Great. Dr. Varadarajan is a professor of chemical and biomolecular engineering at the MD Anderson Cancer Center, and he's also the co-founder and CSO of Oravax, that is a new biotech that is developing a proprietary nasal vaccine technology, which I'm sure he'll tell us a little bit more about. And he'll be telling us about different types of vaccines and how they can be administered. So please go ahead.
If I may, I'm actually the MD Anderson professor, but I'm at the University of Houston. I'm not at the MD Anderson Cancer.
Okay. The MD Anderson professor, sorry.
Thank you. And even though we have a vaccine in the pipeline, I didn't prepare slides because this was an informal discussion. And I'm going to talk about the different modes of administration generally. I'll take this opportunity to remind everybody once again that vaccines have been the greatest invention of mankind. We save about 6 million lives annually worldwide. It has had a better impact on humanity than just about any technological innovation, including Facebook, which we are on right now, and Twitter. So it's important to recognize that vaccines have dramatically changed the quality of life expectancy, and typically they take a while to develop, right? They take about 4 years, sometimes 10 years to develop a vaccine, and that's by design. Most vaccine development there is a heavy emphasis on safety. All of the regulatory agencies want to see that the vaccine is safe before they will release it out to the public. Whereas in our case, depending on how you're counting, the Moderna and the Pfizer and BioNTech vaccines have been ready in essentially 9 months. So there was an article in the New Yorker, for example, a couple of days ago where they said, the Moderna vaccine was formulated in January. They've made very few changes since January, right? What we have that's being considered for EUA, emergency authorization, is really the same formulation. And the reason for that is we have to go through this extensive period of safety testing and efficacy, which is what they've done at record speed over the last 9 to 10 months. Now, there's an advantage and disadvantage of that, right? Conventional vaccines, when you're not in the middle of a pandemic, you can afford to roll out the vaccine slowly. You finish your phase 3 trials with 30,000 people, then you enroll 50,000, 100,000, and you're able to ramp it up slowly and include more and more cohorts of people and see how they react to the vaccine. Whereas in the middle of the pandemic, unfortunately, we don't have that luxury and we want it released right now. And so, there's a question of prioritization. Right now, the vaccine is being considered for people who are considered at highest risk and consequently would benefit the most. But, I'm sure that all of you have seen early reports coming out of UK, for example, saying that people who have, severe allergies, who are prone to anaphylaxis should not be given the vaccine. There's some reports of facial paralysis associated with the vaccine and so on. It's very early days. It's been tested only in 30,000 people. 30,000 people is either a lot or a really small number, depending on how you look at it. In the context of 7 billion people, which is the world population, 30,000 is pretty close to zero. So as we test it in more and more people, there will be more of these coming up. So we don't know the full profile of the vaccines that are coming out yet, but all of the data available to date promises that it is extraordinarily safe and there's a high level of efficacy associated with it. And at least in the US, with the EUA authorization, they will continue to monitor the durability and the safety of the vaccines. It's not like the FDA is going to suddenly stop and say, this is it. The vaccines are ready, everybody get it, we don't care about you anymore. Even for approved vaccines, the DHS and the FDA continue to monitor it. You've seen these handouts as you or your children go get vaccines. It's a continuous process of monitoring of the safety and efficacy of the vaccine. I'll use that to transition to one question that a lot of us frequently think about, which is, if the typical vaccine development takes about 4 to 10 years, how have they been able to develop this in 9 months? Have corners been cut? Is there a political pressure? What's going on here? This seems too good to be true. I don't want to take the vaccine. Very common sentiment. And the answer to that is I will push back on the fundamental statement that we've developed it in 9 months. I will argue that it has taken us probably 15 years to develop this vaccine. Why do I say that? In this purely from the perspective of vaccination, I think we got lucky. What I mean by that is, and I'm very cognizant of the fact that we've had 1.6 million deaths, 70 million people infected. So lucky just in the perspective of the vaccine stuff, that it is almost a second version of the original SARS-CoV-2, right? So original SARS-CoV, which infected, which was born in China in 2002, and then it died out. Now, what happened was because we had that information and we've continued to build vaccine platforms in the interim, the first case of COVID-19 was November, December in China. And then we got the genome sequence on January 6th, which again is in record time. Now, what happened in these 15 years is next generation sequencing worked so well that we're able to go to a genome so quickly. So immediately after you have a pathogen, we're able to get the whole genome sequence. Now, because the NIH and agencies worldwide have invested money in holding sequences of pathogens in databases, they immediately took that sequence and put it into the database, and it told us that it was a variant of the original SARS coronavirus. Now, that allowed us to lean on a lot of the learnings associated with the original SARS coronavirus. Dr. Salazar just showed you the spike protein. It's very similar to the spike protein that was in SARS coronavirus. And so then we immediately knew, based on our learnings from the original SARS coronavirus, that we needed to target the spike protein, which is a trimer, which is the schematic he showed. So a lot of learnings over the years have helped us. And in that sense, it became a vaccinatable pathogen. And consequently, we are benefiting from those years of learning. And so we should really think about it as if we've spent 10, 15 years doing this, not just 9 months. At the same time, vaccine technologies like the one from BioNTech, Pfizer, or Moderna, mRNA-based technologies don't use viruses anymore. And I know one of our speakers is going to talk about the mRNA technology, but again, the fact that we don't have to use live attenuated vaccines, and these developments happen in parallel, primarily in oncology as well, gave us great confidence, right? That we know that these vaccine platforms can be brought here, and you don't have to have headaches associated with the polio live attenuated vaccine, right? So learnings from there helped us as well. The last bit that I will talk about before we transition to the next speaker is, the efficacy of vaccines and vaccines working. What does it mean that the vaccines work? We've all seen numbers, 95%, 94.1%, 90%, and so on. So if you think about it from a vaccine perspective, you can think of 5 metrics at least, right? Does it prevent deaths? Does it prevent hospitalization? Does it prevent clinical symptoms? Does it prevent transmission? Does it prevent infection? Ideally, you want one that covers everything. But in the context of a pandemic, you clearly want to stop the deaths, hospitalizations, and clinical symptoms. Top 3 because they have the huge impact on humans. Now, if you look at the way the current vaccines have been designed, the ones from Pfizer and BioNTech, they're designed to give you a shot in your muscle. And they primarily elicit what is known as systemic immunity. So they prevent hospitalization, clinical symptoms, and deaths. There's no immunological reason or no scientific reason to think, and we're pretty much sure that they're not going to stop infections, right? So people can still have local viral infections in their nostrils. And this is important because even after people get their vaccines, your masks are going to be important so that you don't spread the virus unknowingly to immunocompromised people. The last aspect is the side effects of vaccines, right? So we've seen numbers like 1 in 100,000, 1 in 50,000. Statistics is good, but a lot of people believe statistics is just a bunch of lies, right? Even if it is 1 in 50,000, I don't want to be that 1 in 50,000, right? I want to know that I am safe. I don't care about everybody else. So I need to know it's safe for me. And so for everybody, the key question to ask is the risk benefit of vaccine versus exposing yourself to infection. Now, if you're socially distanced, you're able to wear masks the whole time, you're financially stable, you can work from home the whole time, you have no pre-existing conditions, you're at low risk, and you're not around immunocompromised people, then maybe you can afford skipping the vaccine and say, my risk is so low, I don't have to worry about it. But for everybody else, I would think that, the vaccine would make a lot of sense, even if it is bridging to a more durable immunity. We have lots of questions coming up. But in comparing the risk versus benefit, for most people, it would be beneficial. The worst vaccines we develop are the vaccines that sit on the shelf. They have 0% efficacy if they're sitting on the shelf. They can only work if people take them and we can be protected as a society. And I'll pause that and transition to the next speaker so that we can come back to questions at the end.
Thank you very much. I know you've preempted a few of the questions we had already, and I'm definitely glad that you immediately went for the big question of how did we manage to get here so fast, and that was really fascinating to hear. In terms of the efficacy, I know that those graphs really look incredibly impressive to me. So. Yeah, we'll have some more questions on that in a little bit. But before that, I'm going to then move on to our next speaker, Dr. Jamie Catanese, who is an assistant teaching professor of biosciences at Rice University in Houston. He's also co-founder of another company called Twister Biotech that that is a safe and effective platform for getting DNA into difficult to transfect cells. But I know that's not necessarily what you're going to talk about today. You're going to give us a little bit more detail about what an mRNA vaccine is and how they work. So please take it away.
All right. Let me share my PowerPoint here and get there we go. All right, so I'm going to talk today about mRNA vaccines against COVID. And so just to remind everybody what a vaccine is, we've known literally for centuries, it's already been talked about a little bit here, right, that you expose the body to something and the body recognizes it, it reacts to it, and you build in this immunity, you build this resistance so that when you see it again, you're able to fight it. And when I think about, how amazing our immune systems are, this is biological warfare. We are so grossly outnumbered. I mean, it's ridiculous, right? We live on a bacterial and a microbial planet. Just by sheer number of individuals and diversity of species, we think we're the top of the food chain, if you will, right? But it's ridiculous how many more of them there are in viruses and bacteria and fungi. And I always like to use the example of the Trojan horse. Because this is how our bodies see the outside world, if you will, and that all of those pathogens, that I'm showing here, actually, let me get my laser pointer there. There we go. All these pathogens, they come in different shapes and sizes, but we don't see what's on the inside, right? We don't see the Greek warriors. We just see the horse. And so in respects to, the coronavirus, it's already been talked about, right, with the spike proteins, which are here in red. our bodies are recognizing this outer shell. All right, and we're making these antibodies to that outer shell, and that is, of course, what is part of our immune response. And so in order to understand how the mRNA vaccine works, I need to tell you a little bit about the traditional ones and how they have been developed. And so again, we have our coronavirus here. If I make it into this cartoon of this mad looking red virus with spikes, right? We talked about the spike protein. The traditional vaccines are where we give you the spike protein, which I'm representing by this red triangle, or a version of the virus that's either been killed or it is weakened in a way where it still looks like the original virus in that, right? It still has these red spikes, but this doesn't cause disease. And so again, your body is able to recognize this outer shell, these proteins, and make an immunity. And so how does this work? And so I need to step back a little bit and teach you about biology again, or remind you about biology and what is called the central dogma. And that is that we have DNA, it gets copied into RNA, and that's used to make protein. And so if we think about the end game, that is, we need this protein, right? This is what our bodies are going to recognize in order to make antibodies. We need to get to where our body sees what I'm going to, it's the red triangle here, or it is the spike protein. And remember about your DNA, right? DNA isn't all of your cells, right? You're made up of trillions of cells, but think about your different organ systems, right? You have brain cells and heart cells, lung cells, skin cells. They all have the same copy of DNA, but they don't all use all of the code here, right? I mean, you imagine that, my skin cells on the top of my head make my hair, right? This is protein, which is great. It would be really weird if all of a sudden the cells that make up my tongue decide to make the same hair, right? That would be really weird. And I'm glad it doesn't do that, right? And so there's these instructions, right, that are within the DNA that say when to make the protein, where to make the protein, how much. And it does this by whenever a cell needs a certain protein, and this is happening literally thousands of times every day in your cells, right? It's happening right now. There's this enzyme called RNA polymerase, and it makes a temporary copy of the gene that encodes for the protein that you want, right? And so this is that messenger RNA. This is the mRNA, which is the vaccine that we're going to talk about. And then this temporary copy is read by another biological machine, which is called the ribosome. And that's what makes the protein. And so to turn this into another analogy, anyone who knows me knows I like food. And so I'm going to use a food analogy. And so you're in a restaurant, right? You're the cell and you want a cheeseburger, right? So we're going to make the cheeseburger protein today. And I apologize if anyone is hungry. You're going to look at the menu, right? And in the menu, and I'm showing you it here actually as a book, because in your cell, you literally have tens of thousands of recipes, right? It is a book. All of the proteins that your body needs to make are in this book. And you're going to find the cheeseburger recipe and say, that's the one I want, right? And so then you're going to tell the waiter, which is the RNA polymerase, and they're going to transcribe the recipe that is just for the cheeseburger on this temporary piece of paper, right? And then that's what gets handed to the chef. The chef is the ribosome, and they're going to make the cheeseburger for you, right? So this is the message. This is the instructions to make just that cheeseburger, to just make the protein. And so we're just one step back, right? Instead of giving you the protein, I'm giving you the message so that your body will make the protein. But it's not quite that simple in that RNA, as I have mentioned, is fragile. It is chemically unstable and it is degraded really easy. It's destroyed really easy in the environment and in your body. It's meant to be temporary. And so what's been alluded to is that Pfizer and Moderna and other companies who are developing these type of technologies have to protect the RNA. If I just inject the messenger RNA into your body, it's literally destroyed in a couple of minutes. It doesn't do you any good. So we've got it protected, and they've been protecting it with what's called a lipid nanoparticle, which you can think of as like a soap bubble. And that just allows it to last long enough so that it can get into some of your cells. And so when it gets into some of your cells, then it just releases the mRNA. Well, this messenger RNA looks just like all of your messenger RNAs, right? There's no difference between this message and all of the thousands of messenger RNAs that your cells are making in order to make protein. So the ribosome sees it and it makes protein, right? Everything is natural at this point, right? And of course, instead of this red triangle, it really is the spike protein. And I'm simplifying it here. There's lots of steps, but you're just going to make lots and lots and lots of more proteins. And eventually your body sees it and makes those antibodies. And so that's what that is, in a sense, what the mRNA vaccine is doing for us. We're giving your body, you're giving your cells the message so that it can make the protein. And so with any technology, there are pros and cons. And so one pro with the mRNA vaccine technology is that it is faster and easier to make than these traditional proteins, which is good because the current limitations are that we need 2 shots, right? So we've got to make lots and lots of doses. I've already alluded to that it has a short time span, it's short lifespan. It's also naturally destroyed, right? This is good, right? When you get that shot in your arm, it's only going to last in your body for a few days. And so that's good. You're not going to keep it forever, but it's a double-edged sword, right? It doesn't last that long. It is chemically unstable, which is why you've heard in the news about the ultra cold temperatures that the Pfizer vaccine needs. Moderna is just a regular freezer. This is related to the nanoparticles. But that is a consideration. It is a limitation of this current technology in its current form. It's also been alluded about the side effects. What we know from the data is that the messenger RNA itself doesn't seem to be contributing that much to the side effects, that it is the nanoparticles that are doing it. And you can imagine there's lots of research in this, right? Just as alluded by the previous speaker, right? We've been working on this easily for 10 to 15 years. And lots of work has been doing in this. And there's lots of material scientists and nanochemists who are working on different formulations, right? To where we can get it down to 1 shot, where we could maybe store it at room temperature, where we don't have the side effects, right? There's lots of things that can go into improving these type of vaccines. I put here, and I put it in parentheses because it's technically not an advance over traditional vaccines, but I just want to make sure it's understood that you can deliver multiple messenger RNAs. And the reason it's not an advance is because if you think about the flu shot that hopefully you've done in the last few months, it protects you against 3 or 4 different strains, right? So that kind of advantage is still there. But I put this here so that we haven't lost this capability. We can give your cells multiple messenger RNAs. So as additional variants of the coronavirus spike protein is found, as the virus mutates, we can keep up with this arms race, if you will, right, to help vaccinate. And what I hope I've convinced you, or as you're thinking forward really, is it's really any sequence, right? This pandemic has really, COVID's been the catalyst, right, to get this kind of technology up for, to the forefront, if you will. But after we get this handle on this pandemic, it really can be a vaccine for anything. It could be a vaccine against the flu. We might make a better vaccine against the flu, which has, efficacies of 40 to 60%, depending on the year, right? That's not that great, right? Considering the 90% that we're seeing with the current formulations with COVID. But it's not just vaccines. It's anything, right? And so this is where I want to leave you with the forward thought that this opens the door even wider for that potential of gene therapy. Any disease, like in diabetes or in cancer, where your body's not making a certain protein or it's making a faulty protein, if I can deliver that message to those cells to make the correct, the good, the functional protein, we can potentially cure, right? Or treat a disease, right? So this is, it's amazing technology, and it's another tool, if you will, in our toolbox. And so I'm very excited. It's a silver lining, if you will, to all this mess and this pandemic that we are currently in. And so I'll leave it at that. And again, thank Taste of Science for inviting me, and I'm happy to take questions, of course, at the end.
Thank you so much. Yes, I'm very excited about the possibilities of gene therapy as well. And I know a lot of people have a lot of questions about exactly how the mRNA vaccines work. And I thought that was a really great and clear way to explain it. And I know we have a few more questions about it later, and we'll get to them right after our last speaker, who I'll go ahead and introduce now, Dr. Jessica Lee, who is a clinician and is an assistant professor at the Department of Internal Medicine at the McGovern Medical School in Houston, and who is involved in running clinical trials for COVID-19. And I know that you're recruiting currently people for clinical trials for a vaccine, and we've left some information on that on our Facebook page and on the event page if people want to go back and find out a little bit more about that. But I'm sure you'll tell us more yourself, and I'm looking forward to hearing what you have to tell us.
Fantastic. So I hope everyone can hear me. You can.
Yes.
Okay. All right, so I'm going to share just a few slides. I was asked to talk a little bit about clinical trials, and I am currently, full disclosure, I am a co-investigator on the AstraZeneca clinical trial. But I do want to walk through some of the basics of sort of how clinical trials are formed and the safety behind them, and things like informed consent. So just a quick background, many of you know that Operation Warp Speed is essentially how so many of these vaccines are getting tested and put through clinical trials in a much faster manner than they usually are. I want to emphasize that it's not due to compromising on safety, but due to being able to coordinate things on a bigger scale and in a faster way. So instead of having certain steps that used to occur one after the other, many of those steps and much of it is bureaucratic steps are happening kind of all at once. So it's essentially going through the U.S. Department of Health and Human Services. The goal is to produce and deliver 300 million doses of safe and effective vaccines. And essentially just accelerate any kind of development, manufacturing, distribution of vaccines, and as well as other therapeutics and diagnostics for COVID-19. And to this end, there has been approximately $10 billion of supplemental funding to try to get this done. So when you plan a clinical trial, oftentimes you're using previous data, perhaps from cell studies or animal studies, and then you start developing questions. So who gets to participate in these studies? What kind of people are you looking for? How long will the study last? Is there going to be a control group to try to control for bias? Is it a drug or an intervention that will be given to patients? If it's a drug, what kind of dosage should be given? And then also what kind of assessments will be conducted during the trial and what data will be collected. And then what do you do with the data whenever you do get it? So there's quite a few questions that go into planning an effective clinical trial. So many of you have heard that the phase 3 clinical trial and lots of numbers that are kind of thrown out there. And essentially in clinical trials, there are 4 phases. So the first phase is to test an experimental drug or some kind of treatment in a very small group of people. Maybe it's just, a few hundred people. Depending on the intervention, it might be even less than 100 people. And usually you're trying to test in super healthy, generally people who do not have a lot of comorbidities, to try to evaluate the treatment safety, determine maybe a safe dosage range if it is a medication, and then identify any side effects early on. In phase 2, they move to basically a larger group of people. So in this case, maybe you have a few thousand people, not quite in the 30s to 40,000s that we're seeing with our current phase 3 clinical trials, but a few thousand people to again look further at efficacy and safety. And in phase 3, then you move to really large groups of people. So this could be, tens of thousands of people. In this case, many times you're opening up your type of population to bigger groups. So they don't have to be only super healthy young people. These can be patients who have comorbidities, older participants, that type of thing. And again, it goes back to confirming effectiveness of the doses in a larger group of people, monitoring for side effects. And when you get large enough numbers, you can start to compare to, any of the commonly used treatments. So this speaks more to if you're looking at, let's say, a new treatment for some kind of cancer, and you can compare it to the standard treatment for that type of cancer. And then always through all the phases, they're collecting a lot of information on safety. So most of the trials that are out now for the COVID-19 vaccines are now in phase 3. So they're looking for anywhere from 30 to 50,000 people to be in these trials. Once one of them gets approved, so we found out that yesterday that Pfizer's vaccine was approved for emergency use authorization, then they're kind of basically in sort of the phase 4 type of phase where treatment has been approved for use by the FDA. But again, as one of the other speakers mentioned, safety data still gets collected, even post-marketing. They do a lot of post-marketing studies. They're still looking at risks. They're still looking at side effects. They're still looking at benefits. They're still looking at, what is the best way to use this particular treatment. So also just to kind of highlight, in terms of safety, all of these protocols, any kind of research that is actually ever done, even if it's animal studies, have to go through an institutional review board for approval before the project can even start. Boards are generally comprised of at least 5 members. They'll include scientists, they include doctors, they do include lay people oftentimes. And basically those people are there to ensure that people are taking the right steps to protect the welfare and the safety of the participants who are going to be in these research projects. And they'll send them, and they do it with a lot of scrutiny, and they'll send it back to you if if there is something that they question in terms of safety. On the individual participant side, there's also something or a concept that's known as conformed consent. So usually for research participants who have done studies before, they get a very long document, can be many, many, many pages. It could be shorter too, depending on the type of research study. But it basically will inform a potential participant what kind of project that it is, what are the risks and benefits when they do it, what are they, what are the investigators looking for and the outcomes, how long are they going to be in the study, and all of that is designed to be listed out in great detail so that a participant, they are truly volunteering for the study and not being coerced into it, for some other reason. And, just also a caveat for an individual person who is participating, there may be some personal treatment benefit. Again, for example, in these vaccine trials, if you happen to get the vaccine and not the placebo, you might get some benefit from that. But it is with also the understanding that, maybe if you get the placebo, you don't have a benefit from the clinical trial. There is sometimes potentially exposure to unknown risks. Entering into a study could be, the treatment or the whatever the investigation that they're looking at might be a little different from the standard medical practices that the patients, participants are used to. So many of my participants that have come into my study, say that they understand these risks and, they're doing it because they're really trying to help essentially the world. Many of them come into it with the attitude of, if I don't do it, who's going to do it? And so they're, those are the kind of volunteers that we love because they're really doing this because they think that there's going to be globally is going to help a lot of people all over the world. So to that end, for just a quick blip about diversity in clinical trials, sometimes we get trials in which, people are either very highly educated because they're very motivated to participate in these trials, but we're looking for lots of different kinds of people, particularly for these COVID-19 vaccine trials. We know that COVID disproportionately affects those who are older adults and also certain racial and ethnic groups that are minorities. And these are people who sometimes, are a little bit overlooked in clinical trials. And so that's why I wanted to highlight that, having people from many different backgrounds come into these trials is going to be really important. Other sort of special populations that people look for are sometimes pregnant women and children. I will say that currently none of the COVID-19 trials are allowing those populations yet. Hopefully, as they get more safety data, those groups will be included too. And then also the LGBTQ plus population is something that are groups of people that we are also looking for and also have historically been overlooked in clinical trials. So that's kind of my little 10-minute spiel.
That was great. Thank you very much. I mean, that was really a fantastic introduction to clinical trials, and I think we've had a really great sort of comprehensive primer into both therapies being developed, vaccines being developed, the different types of them, and how the clinical trials are designed to make sure we get these in the clinic. And I think, that was a great way to get started and to lead us into all of the questions that we've had in advance for the panel on all of the different aspects that we've discussed. As I mentioned in the beginning, we had a lot of questions. We went through them in the beginning, and we tried to sort of group them in ways that made sense to make sure that we weren't repeating them. And we will try to get to all of them. And I think we'll probably just dive straight into it. And I think we'll start with some of the questions on vaccines, and then we'll sort of migrate to the different themes, but particularly the question of who can receive these vaccines. And I think this is a good place to start because it ties up really well with the end of Dr. Lee's talk just now. There's been a lot of questions that we've received on contraindications for different populations of people potentially receiving the vaccine or not. And sort of basing on from your last slide, one of the questions that we got was exactly on the giving the vaccines to pregnant women and what the data is on that, or potential effects that it could have on the fetus. Do you know about any data on that specifically?
Oh, so I personally do not currently know of any of the trials that are including pregnant women, unfortunately, although they are a really important population. Right now, they're being very strict about only looking at people who are not pregnant, because the effects on the fetus are just unknown.
Mm-hmm. So we don't know yet. I see Dr.
So we really don't know
Varadarajan
yet.
sort of
Yeah.
nodding ahead. So I think you
Yes.
agree with that assessment.
Yes.
Okay, well, I think we'll move on because we have a lot of specific conditions that people asked us about and how they could affect people receiving the vaccine or not. So we can have this almost as a bit of a quick fire round on these. Another thing that we were asked about was specifically on hypertension and heart conditions, and if there's any contraindications of people that have a history of hypertension and heart conditions receiving the vaccine. Is there any data on that or any suggestions that may be a problem?
So in our current trial, I will say that at least for the AstraZeneca trial, we are including all patients who have comorbidity. We are including those who have comorbidity, such as heart conditions or diabetes or hypertension. What we're asking is that those participants just have been stable, essentially medically stable in the last 3 months or so. So, if they're having a flare up of their congestive heart failure, probably not a good idea to participate in a clinical trial. But, if they've been stable on their same medications for months now, and, they've been, they have had no hospitalizations or other acute issues, we are actually absolutely including those participants in our trial.
And so far, there's nothing to suggest that they may have any specific adverse effects to the vaccines.
No, so far we have not had. Yeah, there has not been any evidence that they are affected in a greater way by, say, side effects from a vaccine or something like that. No.
Great, and related to the previous point of pregnant women, another question that we obviously got a lot was the question about kids. I know there were some discussions on that on the panels for the FDA approval of the Pfizer vaccine, specifically on what exact age should you start administering the vaccine. So what do we know in terms of at what age you should start giving the vaccine and whether or not you should give it to kids?
So I
know who wants to jump in on that.
Yeah, I know. Again, in terms of the trials, I don't think that they've had anyone who is younger than the age of 18. But if I'm not mistaken, the EUA for Pfizer that came out is actually 16 and up. So they are including some younger. They're just not including very young children, but they are including some by definition children, those under the age of 18.
I see Dr. Catanese sort of nodding as well. I don't know if there was anything else that you wanted to add to it, or
no,
is
that's
just
exactly
agreeing.
right. I'm agreeing. I'm totally agreeing. Yeah, and that's actually one of the groups. They want more data, right? And but we just don't have that data yet.
Well, hopefully we will be getting that data soon. And then obviously the question about people who have actually had COVID-19, and I think the question is to do with mostly people who have had asymptomatic COVID-19 and who may or may not know about it, or what do we know about how much these people may have acquired immunity to the virus, and whether or not that may influence whether some of these people may be included in some of these trials or in receiving the vaccine. I'm not sure how much we know exactly about how much immunity people who have had COVID have and how long it lasts for, specifically if they had it asymptomatically. Is there any data on that? Does anyone want to talk to that specifically? Dr. Salazar?
Yeah, I can chime in on that. Just looking at our donors and the antibody data from our donors, but also the data that Pfizer submitted to the FDA, I think, is also informative. So what we know is that people have variable immune responses to the virus. What we've found from our donor population is that some donors mount a very strong immune response, and some mount almost no detectable immune response at all based on the tests that we have. For example, just looking at the antibody tests, a small minority of our donors, they tended to be patients, younger patients, especially females that had asymptomatic disease. Those seem to have undetectable antibodies. Not all of them, but just a very small proportion of them. And why that's the case, we don't know. Might have to do with viral load, et cetera, how much virus they actually get exposed to, et cetera. What we also found is that those patients with more severe disease had higher antibody levels and a more robust immune response. So that's from our donors. But looking at the data that Pfizer submitted to the FDA, when you review that document, actually a remarkable document showing the data from the vaccine studies, they were both able to study antibody levels, neutralizing antibody levels, the ones that disrupt that spike ACE2 receptor interaction. And they found that the vaccine, the Pfizer vaccine, elicits antibody response similar to convalescent donors, if not better sometimes. So that's actually really encouraging. And they were also able to look at a different part of the immune system, the T cell response. And they found that the vaccine elicits a pretty robust T cell response also. So there are a lot of questions that we don't know about, and it's why it's so important to work with folks like Dr. Lee and with the Pfizer study, because what we have to do is to continue to collect data. The way out of this crisis is with data. And what we're going to have to do is certainly follow the Pfizer population. What they plan to do is over the next 24 months and see, well, how long does this immunity, how long does this 95% efficacy last? Are we going to need booster vaccinations in a year or two? These are questions that we don't know yet, but that we can answer. And that's the important part. And that's why it's critically important that we enroll in studies like Dr. Lee's.
Yeah, and that's you've preempted some of the other questions that we had, which are exactly to do with how long will these vaccines provide immunity for, and whether or not this will be like the flu vaccine where you need to get a booster and a new vaccine every year. And the point is we don't really know the answer to those questions yet. We're just about to start rolling out the vaccine, and it's monitoring the response to the vaccine that will give us the data to be able to answer those questions.
Sure. And what we have seen, actually, there's a really beautiful, absolutely beautiful paper from Stanford in Science yesterday, looking at immune response over time in convalescent humans. And what we know, number one, is the test that you do matters. But in general, looking at their data and our data and data from other people across the world, is that these antibody levels seem to wane over time. So there's kind of an early response that rises, really nice rise, and then it kind of seems to plateau and then wanes over time. When we look at those specific antibodies that neutralize a virus, we know that the very high levels come down, and especially after 60 days, you don't have those very high levels. But what we don't know is what's the threshold, and what is the exact biomarker that tells you you have protection. Nobody really knows that yet. We think we kind of understand it, but we don't know that yet.
Great. Okay, so back to the question of who can receive the vaccine. There's been a lot of news in the media recently about allergic reactions and the possibility of people with allergies having adverse reactions to the vaccine. And I was wondering if you could tell us a little bit more about that. Specifically, I think there were some data from the UK that suggested that people with allergies were recommended not to get the vaccine. What exactly is the reason for that? And are there specific allergies that people should be wary of?
So I think in relation to the University of Oxford study, I think what the concerns were about the allergies are, vaccines are made of different components, and depending on which one is produced by Pfizer or Moderna or any of the other ones, they each have a little bit of different components to them. So if you've had a history of having allergies to previous vaccines, you may be allergic not so much to the vaccine itself, but to a component of the vaccine. For example, the very common flu shot, for those who have an egg allergy or a severe egg allergy, it's really not recommended or contraindicated actually for those patients to take the flu shot. So I think it's more along those lines. I don't think there was anything that was specific to these COVID-19 vaccines that was some kind of new allergy just specific to these vaccines. I think it's just very typical ones that are seen for other vaccines too.
Yeah, so no normal concerns to have with vaccines. Dr. Salazar, sorry.
Well, I just think it's absolutely critical to point out that these reactions, the ones that were noted in the UK, appear to be incredibly rare. I think that's an absolutely important point to share. When you look at the data that was submitted to the FDA, I did not see a single, report of an anaphylactic reaction in that data. I mean, you see sort of side effects that you might expect with a vaccine, certainly local, pain. I know my arm hurts after a flu vaccine. Some people reported fever, chills, these kinds of reactions. But in general, they're relatively rare. That's one thing. The second thing is with respect to these sorts of local reactions and even systemic reactions like fever or chills, I don't believe anybody in the Pfizer study received prophylactic like Tylenol before getting the vaccine. So that's something that might actually help sort of.
Could you tell us a little bit more about what that is and why it's important?
Yeah, so Tylenol, like an anti-fever medication, antipyretic, something like to prevent fever or chills, or even help with some pain. As far as I understand, nobody in the Pfizer trial received, a prophylactic dose of Tylenol before or after the vaccine. And, that might help actually prevent or treat some of these like local reactions.
That's a great, sorry, Dr. Lee, go ahead.
No, I was going to say, we've been observing the same thing in our study. So, these sort of maybe feeling a little feverish, having a sore arm, they also are very short lasting. So they 24 to 48 hours, those symptoms resolve. And, our trial also has been, pre-explaining to people that they can take over the counter, Tylenol if they need to, if they're experiencing any of these symptoms.
Okay, and this is a great segue to the next point that we had obviously a lot of questions about, which was the existence of side effects. And it sounds like most of the side effects that have been reported are the normal side effects that you expect for something like a flu vaccine. There were some questions about the potential of people with autoimmune disorders having more severe side effects, something like cytokine storms. Is that something that you could speak to a little bit about? I don't know. Dr. Salazar, I saw you nodding, or Dr. Lee, who wants to take that on?
I think it's a tough question because we don't have a lot of data to guide us. And I might defer to Dr. Lee a little bit if she knows, but one can envision a mechanism whereby an autoimmune disease is exacerbated or brought on by a vaccine. Certainly there are rare examples of a vaccine causing an autoimmune condition, like something called, this is going to sound weird, Guillain-Barré disease. which is a sort of an autoimmune disease attacking the nerves and the neuromuscular part of your body such that you develop some weakness. That's pretty rare. It happens, like all vaccines. There are very, very rare signals of an autoimmune condition. I'm not aware of any data to suggest that this specific vaccine exacerbates autoimmunity in any way. And certainly, I'll give you the example of my mother-in-law asked me a question. She said that I heard that the vaccine might make psoriasis worse. And here's my thinking on that. If that were true, which I don't even know if it's true, would you rather have a minor exacerbation of psoriasis versus, severe COVID-19? And I think the 95% efficacy of the vaccine really, that's an easy question to answer. And that's the way I think about it.
Yeah, so far of all of the very mild side effects that people may have seen, like, pain at the site of injection, nothing really has been observed that affects that risk-benefit calculation, basically. And it's still really important to make sure that we get people to take the vaccine.
Not in any way at all. I mean, it's I'm looking at the data, and if you can, I really highly encourage you to Google the FDA document where they share the data, and there's this beautiful curve, which I had never seen before, but this beautiful curve looking at the incidence of COVID-19 after one dose in the placebo group versus the
incredible.
Recipient group, and it's absolutely incredible where you see, like, after about 10 days, the vaccine group levels off at a very low level, and the placebo group just keeps going up and up and up. And wow,
I believe we actually pasted that graph, posted that graph recently on our Taste of Science page, so I think people could go and have a look at that. It's a really incredible piece of data there.
Yes, sir.
And on that note, I guess finally on the side effects, and this is probably quite speculative as well, but there was some news that we saw and that people have specifically asked us about the onset of, I think it was 4 patients that showed symptoms of Bell's palsy that were included in one of the clinical trials. I'm not sure if you're aware of that or if any of you could speak to those particular cases.
I was not aware of the Bell's palsy part, so I'm sorry, I can't.
I did see that. I don't remember if it's noted in the FDA document, but what I also heard is that the rate of Bell's palsy that was noticed amongst the 44,000 participants is the same as the incidence of Bell's palsy in the general population. So again, indicating that it's not a very strong signal
not vaccine related.
Right, exactly, or a very strong signal to suggest that there's a enormous safety concern with respect to Bell's palsy.
Okay, so again, basically stressing out most of the side effects that we see are very, very mild, exactly what we see with most of the vaccines. I guess the only other thing to mention on that note is obviously, to going back to some of the things that Dr. Varadarajan said in his talk about, how fast this vaccine was put into the clinic. People then get concerned about the possibility of long-term effects, specifically in the context of the disease that we know has long-term effects. How can we, I guess, talk to people about those, the possibility that longer term effects may arise that we're not aware of yet?
I mean, that's almost an impossible question to answer, right? I wish we had a straight answer for it, but that's true for every vaccine, every therapeutic that we ever make, right? How do you know something bad's not going to happen in 10 years? We can only rely on what's known, and unless there's a specific reason to suspect it, it's unlikely. But again, I don't have a rational way of answering that question, unfortunately.
Yeah, we're making educated guesses for this, and so while they are educated, there is still the guess component, and so I think it's really hard for us to be able to to define that part.
I guess it goes back to the whole concept of risk benefit again, right? And right now there's much bigger benefits in getting people vaccinated and not much evidence that it will cause harm.
I do think that there's a rational way to sort of think about the limited data, and that really has to do with what Dr. Catanese or Jamie mentioned in terms of the and I apologize if I butchered your last name, the composition of the vaccine. It's a lipid nanoparticle with mRNA. Number one, mRNA doesn't last very long at all. I mean, it's going to get degraded pretty quick. So how long does that lipid nanoparticle last? Probably not long either. It probably gets degraded or digested, or some kind of breakdown occurs. So what's the likelihood that we're going to see side effects past the 2-month period? In my opinion, it's probably relatively low, and that's why the FDA would go ahead and act on a 2-month safety follow-up, which I think is probably pretty reasonable. That's not to say that there might not be some other signal that emerges, but strikes me as very unlikely.
Great. Okay, I'm very much aware of the passage of time, so I think we'll move on to the next topic that we had a lot of questions about, which was just the logistics of the rollout of the vaccination and the vaccination campaign itself. And obviously a lot has been written of the fact that these mRNA vaccines are administered in 2 doses. So a couple of the questions that we had, which I guess I will sort of put together into one, is number one, what happens if someone gets sick, or is it possible between dose number 1 and dose number 2? And what are the consequences of someone not getting the second dose at the prescribed time after the first? And any volunteers to take up any of these questions?
I think I don't know the answer to what happens if you get sick between dose 1 and 2. I know that in the Pfizer data, they collected that data, and they did see some people that got sick between dose 1 and dose 2. In reviewing the data, most of those occurred before day 10, especially, and certainly before day 14 is when most of those cases occurred. After day 14, it seems like you've got about what they claimed was about 80% efficacy. It looked like, or it ranged somewhere between 50% and 80%, depending on how you chop up the data, I think. And so again, looking at that curve that I mentioned, even after 1 dose, you've got pretty good protection, especially after about day 14. Now, whether there's a rule that says you can't have the second dose if you get sick between the dose 1 and 2, I'm not aware of that. I don't know, Jenny, Jessica, is that something that you've seen?
No, yeah. So like in our trial, again, we've had, a few participants that experienced the sort of the vaccine reaction after the first day, after the first injection. Everyone had their symptoms resolve relatively quickly. And so we have not yet had anybody who has not gotten the second dose. And interestingly enough, several of our participants who initially did have some reaction did not have any reaction really after the second dose. So there's currently no clinical evidence to prevent someone from getting a second dose if they have a vaccine, what we consider a vaccine reaction after the first dose.
Dr. Varadarajan, I think you were trying to say something as well.
From an immunological perspective, both animal and human data support that the second shot just boosts the durability of the antibodies that are elicited. That, ideally we want everybody to get both shots, right? But it seems like most people should have protection even after the first shot. Now, you might compromise the duration of that immunity if you don't get that second shot. But, if for some reason they absolutely cannot get it, do they have protection in the interim? It seems like the answer for now, based on both human and animal data, the answer should be yes.
Okay, well.
And I will also say that there are actually some trials out there now that will be looking at a single dose, a single dose vaccine. So if you are really curious if one shot works, sign up to volunteer for one of those clinical trials because they are coming down the pipeline for sure. And they will definitely need people to sign up to see if a single dose is effective.
And yes, again, look up the information to sign up for clinical trials if you can, and if you're in an area like Houston where these clinical trials are available. So another question that we got, I think, a couple of times was about whether or not people who get vaccinated have, while not contracting the disease, the ability of actually spreading it. And I think this was a question that was asked at least once in the context specifically of protecting your family members and in giving the vaccine to caregivers that are caring for medically fragile family members who cannot get the vaccine. Do we know about transmission rates from people who are vaccinated? Dr. Varadarajan.
I don't think we know about transmission rates, but again, looking at how the vaccines are designed and administered, your body, or our bodies have different levels of protection, different mechanisms of protection. There's the systemic immunity that's supposed to provide bad infections and takes care of disease, right? But at the point of entry, the nostrils in the case of COVID-19, and the anal compartment in the case of HIV and so on, we have additional protection that prevents viral entry at the first point. And none of the vaccines, the Pfizer and Moderna vaccines do not elicit vaccine at the point of entry. So if we have to make an educated guess, and the data in the animal supports this, that they do not prevent viral seeding in the nostrils. They will make sure that you don't get sick and end up in the hospital. And that was the information that was tracked upon vaccination. I think the AstraZeneca trial is different because they do the PCR testing as well. But for the Moderna and Pfizer vaccines, it's likely that people can still carry mild viral loads in their nostrils because it's not specifically designed to prevent that.
Mm-hmm. And those viral loads, we don't know if they're sufficient to actually be transmissible.
They're likely to be low, and if you have an immunocompromised person, in the context specifically of an immunocompromised person, yes, they will be able to transmit. And so this is why I personally think it's still important until a large fraction of the population gets vaccinated, that even if we get vaccines and we get vaccinated, that we should continue to wear masks, just as an abundance of
social distance.
Yeah,
absolutely. That's really good advice. Are there plans to test, I guess, this is really on the logistics of the vaccine rollout and the vaccine deployment. How will it work? Are there plans to test people for COVID vaccine before vaccinating, or to test for immunity and antibodies post the vaccine? Is any of this included in the rollout plans, or is this not something that is currently predicted?
So yeah, so I'm in the Department of Pathology, and we have the various tests available, and at Methodist, at least, there's no plan to test for antibodies before the vaccine. And certainly the recommendation that I've heard from all the experts is even if you previously had COVID, to go ahead and get the vaccine anyway. So I don't think that a test before the vaccine is going to help us much. The question that I have is, should we test after the vaccine? And so we actually just started doing this test on Friday. We rolled out a quantitative test, meaning an antibody test that actually gives you a level, tells you like how much of an antibody you have. And that's something that might be worthwhile. Like if you get vaccinated and you get tested with this antibody level, and the level is 0, or you don't have any antibody at all, that might be informative. I mean, I don't know that yet, but that's something that I would certainly, want to know.
Sorry, I muted myself, but yes, that's a really good point. I think there's Dr. Catanese who mentioned this in his talk about, there's a lot that has been written about the storage conditions that we need to keep some of these vaccines on, and specifically for these vaccines, I think it was for the Pfizer vaccine, it's been reported that each box of the vaccine has about 800 to 900 doses. How much do we know about how long we can keep these vaccines for? And do they kind of all have to be administered pretty quickly after they're sort of taken out? Do we know that?
Right. Yeah. So my understanding is that it's a few days, like 4 or 5 days in the refrigerator once that vial is thawed. And so that is in some respects, it's just the logistics of make sure you have your 800 people lined up that you know you can give them those shots within the 4 to 5 days. That's my understanding. You know, as far as the how long it stays good or viable after it's been thawed. So that in a sense is like, you know, don't take all the vials out of the freezer, right? Take them only out as you need them. And but again, it's just logistics. It's just, you know, there's smarter people than I am as far as logistics. I'm glad I'm not the person in charge of that, because it's to me, I would say a nightmare just thinking about all the different things that have to go on to ensure people are getting the shot when it's still good, and then thinking about that now they've got to do it 3 weeks later as well, and that's the challenge.
Big logistical challenge for sure. I mean, there were a couple of people who just asked generally, and I guess I'll just take your cue for that, what your concerns are in terms of the logistics of the distribution of these vaccines. And I don't know if there's anything that you wanted to talk to specifically on that, on the follow-up from this.
I would say I personally don't have, like, you know, major concerns in the sense of, like, I'm not in charge at all of, like, who, you know, which states get how many shots or, you know, many doses and how those are being distributed, because that's on a state by state level. You know, like I said, that they're smarter people than I who are doing, you know, this distribution, and it's a matter of making sure you've got the doses, you've got the PPE for the folks who are actually administering it. You know, but other than that, I don't have any concerns outside that.
So for Texas specifically, since you're all based in Houston, what do we know what the Texas plan is for who will receive the vaccinations first? How does this work in terms of how each state determines its own plan? You've just mentioned these are state-based decisions. This is not a federal decision, but I guess specifically in Texas, what do we know about how the rollout is going to occur? I guess this is probably more of a question for Dr. Lee.
Yeah, so recently the CDC actually put out their recommendations sort of who will receive vaccine first, and they kind of categorized it as 1A, 1B, and 1C. 1A is essentially healthcare workers who have direct contact with COVID patients, as well as nursing home patients. And so logistically, many of the medical centers, because they have -80 freezers, will actually be doing that. And then I believe I read that there's a plan for like Walgreens and CVS to provide the nursing home patients with, since they also have the capabilities of storing. 1B is supposed to be also healthcare workers, but those who are more ancillary, so perhaps not direct, care of COVID patients, but care of patients. And so that's kind of like the 1B group. 1C, my understanding is older adults actually who have comorbidities, perhaps younger adults with comorbidities, but I think they're focusing currently on older adults with comorbidities. So that's my understanding of who's going to sort of get rolled out first. So because Pfizer and Moderna, can only produce so much vaccine doses right now, I think most of those will probably end up getting used in healthcare settings or nursing homes. And further, other vaccines that are coming out, such as AstraZeneca's vaccine, there's the Janssen's vaccine, those kinds of things, will probably be for later distribution for more of the general public, because they'll also be able to produce an extra number of doses. Yeah.
Great, thank you very much. So we had a question that I think we've had a couple of times that I understand is probably going to ask for a little bit of speculation on your part, but bear with us. Particularly, you know, in the context of the importance of making sure people understand the importance of vaccination, and in the context of the existence of a bit of skepticism. What do you think the next few months to 2 years look like if, say, a quarter of the population gets a vaccine versus half the population get a vaccine versus 3/4 of the population get the vaccine? The amount of how will the amount of people getting the vaccine affect how our lives will be from now on? And I noticed Dr. Varadarajan sort of nodding ahead as I was mentioning this, so I guess I'll start with you.
No, again, right, it's very hard to convince people who are firm anti-vaxxers that they should get the vaccine. But I think, you know, there's also a lot at stake, because if we get this wrong and we trip on some safety, it's going to hit all vaccines, right? People won't just remember mistakes with the COVID-19 vaccines. They'll remember it as mistakes with vaccination. And it will quickly erode the public's trust in vaccination as a whole, not just, I won't get another COVID-19 vaccine. I just don't want to get another vaccine. There's more data to support that. So, you know, ideally, the 25, 50% and so on assumes that we have enough manufacturing, and nothing at this point suggests that we have enough manufacturing to cover the entire US population within the next 3 months or even 6 months. It's likely to take longer than that, just because the vaccines have to get manufactured, distributed, and so on. And like Dr. Lee mentioned, the CDC has very nice set of guidelines that hopefully, you know, the Texas and all the other states will implement in how to prioritize people based on their risk profiles for getting COVID-19 infections. So it's going to take a while before all of us get vaccinated, and that's assuming everybody wants to get vaccinated. And so a related question on herd immunity also depends on, you know, completely getting rid of infections in the nostril and the upper respiratory compartment. If you're not able to get rid of it completely, then the fact that these people can still seed infections even at low levels to other immunocompromised people might be a concern.
I don't know if anyone else has anything they'd like to add as well. If not, then I can go ahead and move on to the next set of questions. And there was one question that we got, I think that was more directed towards Dr. Lee. And I think you mentioned specifically the importance of diversity in clinical trials. And there was a question on specifically the issues, the historical issues with marginalized people either having difficulty in receiving the vaccines, or, you know, in the case of the Tuskegee, being skeptical of getting vaccines because of some historical issues that happened there. Are you aware of any efforts to help recruit members of these specific communities for vaccines and for the trials?
Yes, I will say that all of the trials, you know, from Pfizer, Moderna, from AstraZeneca, they have all been very, very interested in recruiting people from minority populations, people who, again, are disproportionately affected by COVID, but may be underrepresented in trials. And so there's been a lot of outreach. There's been a lot of actually education for us as the investigators on how to reach those populations, you know, educational materials that we can potentially use. I had a brief conversation with someone who was mentioning that, for example, in the Houston African-American communities, it might be best to talk individually to their doctors or to their churches, because those are the people that they really trust. And so we are going to plan to start to make some efforts to go out to do some outreach in those areas specifically, because they might not be reached by radio or television or, you know, newspaper, which is some of the typical ways that we'll recruit people. And so we have had many presentations and conversations about how we can better reach those that we really need to have more representation for in these trials.
That's great. Thank you very much.
Mm-hmm.
On a different question, picking up from some of what Dr. Varadarajan mentioned in his talk, and the fact that yes, the development of this vaccine was very quick, but it was also the culmination of 15 years of research on a lot of different aspects. What does this mean in terms of what we can expect with vaccine development going forward? Is this how vaccine development is going to look like from now on? That we have a new disease that we want to build a vaccine from, we can get it done in a few months, or was this something that was specific to this case, or is that where the science is right now? Could you speak a little bit to that, please?
I think it's a combination of two different things, right? In this case, like I mentioned, there was original SARS coronavirus, so we were able to map the genome, align it, identify the proteins, the spike protein primers, able to design it quickly, mRNA technologies. But that's why I said from a vaccination perspective or vaccinology perspective, we got somewhat lucky, right? That the virus also turned out to be vaccinatable, right? If that's maybe I just made up that word, right? But it turns out to be vaccinatable. You can imagine a scenario, and I don't wish this on humanity, myself, or anybody else. Something like HIV, which mutates so quickly. If you have a pathogen with a high mutation rate like HIV that comes in through the respiratory compartment, 30 years we've been trying vaccines against HIV with very limited success, right? And so it's hard to tell, right? If it's closer to something we've seen before, then the mRNA technologies are spectacular. We really like how quickly they've been able to progress and produce a vaccine, that's ready for humans this quickly in 9 months. But at the same time, we have to be cautiously optimistic and continue to, think about vaccine developments, newer platforms, whether it's the AstraZeneca's, adenovirally vectored vaccines, different platforms. It's good to have many different arrows in your quiver to throw at it. If all of them work, we're all happy, spectacular. But I think we should not lose sight of the fact that we will need diverse platforms to be able to tackle the challenge. And just because it worked at the first shot here, or it seems like it is going to work, right? Again, being cautious, it seems like it is going to work at the first shot here. This is not to immediately jump to the conclusion that vaccine development suddenly has such a, accelerated timescale in the future. Hopefully, reason for optimism, but again, reason for caution as well.
Optimism and caution. I think those are two really good points to do that. Yes, sorry, go ahead.
I was just going to add something also that was said earlier, and I'm trying to remember who said it, wasn't me, but about how, with this particular situation with the pandemic, that, a lot of the bureaucratic hurdles have been removed, not the safety, right, but the bureaucratic handles. And going forward, if it's not a pandemic per se, those are probably still going to be there. And so it will slow, in sense, I'll say the deployment, maybe not necessarily the development, but the deployment of that vaccine. But again, this is just like, with the mRNA vaccines, as I said, like, it's another tool, and there's more tools, lots of tools.
Yeah, and it's great that we've got so many at our disposal, and I know that we're getting very, very close to time, so I'm just going to try and get through a couple of more maybe more specific questions before we get to our time. And one of the questions that we had, obviously there was a lot written about the Oxford AstraZeneca vaccine and how a lower dose seemed to have a better result than the higher dose. What was, I think Dr. Lee might be the right person to ask about that. Could you explain a little bit about that?
I'll explain a little bit. So I've actually had a lot of questions about that from participants.
I figured you probably did, yes.
So I think the thing that we have to remember is that the low dose was actually a mistake. And so it was an accident that they happened to put a first lower dose and then the second regular dose in what is actually a very small number of people. So I want to be very careful about drawing any conclusions about efficacy or things like that, because the number of people that received that lower dose was so small compared to the number of people who have received both full doses that, it would again hesitate to make any real conclusions about that. That being said, I do think that AstraZeneca may in the future plan for some well set up or better set up trials, not a mistake, to see if there is some truth to the low dose first and the full dose second. But currently there are no plans to change any of the trials that are running right now into a half dose, full dose. So just, remembering the very small sample size. So, if we're looking at phase 1, 2, and 3, that would have been in phase 1 to 2-ish. So, I really wouldn't make any conclusions out of that.
Fair enough. On those phases, one of the questions that we actually got during the event was whether there will be a phase 4 for the vaccines with emergency approval.
Yes, absolutely. Yes. The FDA will be sure to be monitoring all of these vaccines very closely post-market. And so, and remembering too that an EUA is emergency use authorization for a reason. It is supposed to only be used in cases where, the risk of getting COVID is so much higher than the vaccine that it's well worth that risk, that an EUA doesn't actually apply to the general public. And so, there will most certainly be monitoring during the EUA that's occurring.
Great. So there's a couple more questions, I think, on the logistics that I'd like to go through before we close. And one was on the FDA on these emergency conditions, allowing vaccine manufacturers to skip pre-approval inspections of their facilities. And they wanted to ask, what do we feel comfortable with that from a manufacturing standpoint? And if they're not necessary, why use them in the first place? That's quite a specific question, but still would like to hear your thoughts on it. Dr. Salazar, maybe?
Well, so the question was about pre-approval inspection of the facilities for manufacturing.
Of facilities for manufacturing of the vaccine, yes.
Yeah, and I think this actually speaks to what I think Dr. Catanese mentioned about bureaucracy and trying to clear any hurdles to getting needles in arms, getting the vaccine in susceptible people. And my suspicion is that it's along those lines. And so, if the vaccine that was used in the studies was manufactured in those facilities, and we've not seen any major adverse events as a result of that, clearly no systemic infections, no contamination as far as I know of any of the vaccine doses in the study, then sort of the study, I guess, in this context, and in the context of an EUA, takes care of some of that concern. Now, why can't we do that? The question was, is this stuff even needed in the first place? And I think it's probably a mistake to extrapolate the current situation to normal operations. Number one, because this is a very high profile study with a lot of eyes on it, so there's not a lot of room to make mistakes, i.e. putting half dose by accident in some vials, that's going to get known. And then there's going to be a lot of people looking at that and saying, why did that happen? So there's not a lot of room for mistakes. But under normal circumstances, that's not the case. And not all companies manufacturing drugs are Pfizer's, and they're not all AstraZeneca's. There's smaller companies that maybe don't have the expertise that these big drug companies have. And so we absolutely have to maintain quality control, maintain these types of inspections and accreditation. For example, one of my jobs is to inspect blood banks and labs across the country to accredit them to indicate that they are performing in a safe environment and performing well. And that's such a key part of our safety mechanisms, our checks and balances that we have in healthcare to absolutely provide the safest and best healthcare that we possibly can. And so that's what we have to do.
I think that was very clear, and on the case of COVID, obviously, we wanted, as you mentioned, to get back needles in arms as soon as possible, and we were able to push that and still make sure that there were no adverse effects related to that. And as a last question related to that, and sort of differences that were you know, associated with using the emergency use authorization to get these needles and arms as soon as possible. Was there any other changes to normal protocols from the pre-COVID era in terms of how these trials were processed, in terms of the amount of patients being involved, for example, or in any other way? Or was it all was pretty much by the book?
I can take a first stab at that. And in my opinion, number one, not only was it by the book, but in my opinion, the data are even more robust than you might see for other drugs that have approval. I mean, we're talking about 44,000 patients in this study. How many treatments do we in medicine administer that don't have a randomized controlled trial backing them up? I mean, a lot of what we do in medicine is not completely based on a randomized controlled trial. And so what I think one of the good things that's resulted from COVID-19 is that we've got a lot more people educated about evidence-based medicine and about science and about the clinical trials and about the effort that has to be put in place to get that data. And so hopefully there have to be lots of takeaway lessons from COVID-19. And hopefully one of them is that we have to do rigorous controlled studies to be able to move forward in medicine. The way to get out of crises, not just COVID-19, but HIV, cancer, heart disease, you name it, is with data. That's how we get through it. And hopefully that's one of the takeaways that we can have from COVID-19.
Dr. Varadarajan, I see you wanting to add to that.
Yeah, and I think the important thing to remember is, we had an accelerated timeline. So what did the FDA authorize under these special pandemic circumstances that they would not normally do? For example, for the Moderna, they allowed phase 1 to look at safety at the same time as evaluating, more preclinical animal models, looking at things like challenge models. Typically for pathogens, you have to have a pathogen challenge model where you vaccinate the animal with whatever vaccine you want, and you challenge it and show that it confers protection before they let you walk into humans. But in the current pandemic, they said you can do this concurrently with a phase 1 trial, right? Which saved a lot of time. So it's not like we skip the studies, we just have a lot of these happen in parallel, whereas if you did not have a pandemic, you would do them sequentially, again, for safety reasons, right? Safety is always the primary consideration here. Even if you don't vaccinate, it's okay. You cannot vaccinate and cause adverse reactions or worse, kill people because of the vaccine. And so a lot of these happened in parallel as opposed to sequential, which, accelerated the timeline. So we didn't skip those steps, we just did them simultaneously.
And I want to highlight too some of the sort of the more bureaucratic types of things. To run a clinical trial requires a lot of personnel, requires a lot of supplies, things that don't necessarily have to do with the safety of the participant, but you have to order those supplies, they have to ship them out to you, you have to have them available. So for example, all of the PPE I need to wear when I'm, doing a nasal swab on one of the participants, where does that come from? So in this particular case, they put a lot of money and effort into getting these supply chains up and running so that all of these sites around the world could have access to the supplies that they need to run these clinical trials. For example, if they're doing IRB protocols, instead of having a bunch of different ones, just do one big IRB protocol and then have it filter out through to each of the institutions, and each of the institutions approves them concurrently. So those are kinds of things or examples of the procedural types of things that were, that could be shortened, that don't have to do with safety or not looking at, efficacy, but more of the bureaucratic type of stuff.
Mm-hmm. Yeah, and I guess, as we've mentioned a few times before, this is really a testament to what can be achieved when there are no obstacles to the scientific development and to all of these minds being put together to finding solutions to a problem. And I think this is a good optimistic note in which to leave it, because unfortunately we are getting to 10 minutes over time already, and we are very much aware that there's a lot of questions that we didn't manage to get to. We had a lot of questions throughout the event today as well, and we tried to get to as many of them as we could, but we really want to encourage everyone, if you have any questions that we didn't manage to get to, or if you have other questions that you've come up with since in the context of this discussion that you didn't get to send to us, please go ahead and post them to us or send them to us on Facebook, and we'll make sure we, you know, if our experts don't mind, that we'll share them with you and get you an answer to them later on, because we really want to make sure that everyone comes out of this event with answers to all of the questions that they may have to the development of these clinical progresses in terms of both vaccination and therapies. I also, again, want to encourage everyone to go look at what we posted for Dr. Lee's clinical trials, particularly if you're based in Houston. They are recruiting for clinical trials for the AstraZeneca vaccine. So please, if you'd like to take part, go have a look at the details and make sure you apply for it. And on that note, I think as we approach the end of the year, this was a really sort of great optimistic note to look at the light at the end of the tunnel and hopefully you know, get us to the other side of this soon. I want to take the opportunity to first thank every one of our participants who sent us questions that were really fantastic ones that were, you know, the fuel for the discussion that we had today. Obviously, really want to thank our four experts today for taking the time to participate in the roundtable and answer all of these discussions and answer all of these questions and let us know about what they're working on, and the rest of the team at Taste of Science Houston for all of the work that they did in the background to make sure that this event happened seamlessly. And I think as we end this event, you know, there is light at the end of the tunnel, but we are not quite there yet. So please, everyone, wear a mask, continue practicing social distancing, and make sure you vaccinate once you're able to do so. And hopefully we will find ourselves at the other end of this tunnel soon enough. And on that note, thank you everyone for joining us. Happy holidays, and we'll see you on the next Taste of Science event.
Thank you.
Thank you.
Bye.